Hello everyone, this is Divya Jyoti from Vidya Lakshmi. Today we are going to learn about structure of DNA from molecular basis of inheritance. What is DNA? DNA is deoxyribonucleic acid, which is referred to as molecule of life. DNA, what is the structure of it? Actually, how it looks. It is a double helixed structure resembling a twisted ladder. These two strands, it will contain two strands which are held by a chemical bonds with the nitrogen bases. Where is this DNA located? DNA is located inside the cell. Now cell contains nucleus. Nucleus inside the nucleus is the chromosome, and in the chromosome is our DNA. Structure of DNA. These are the 4 key points about structure of DNA which we are going to learn today. Backbone of DNA, which is made up of sugar and phosphate, and nitrogen bases. Nitrogen bases are divided into purines and pyrimidine. We'll learn in detail in the coming pages. Nucleoside, which is sugar and nitrogen bases, and nucleotide, which is sugar, nitrogen base plus phosphate. Pentose sugar. Before going into the detailed structure, let us learn few things about DNA, what it contains. It contains a pentose sugar, a ribose. A ribose is straight chain catenation is this. Ribose is made up of C5, H10 and O5 structure. O5, whose straight chain catenation is here. C1, C2, C3, C4, C5. 5 carbon with its hydroxyl group and hydrogen group here. When that is being brought into a cyclic structure, here is where it is. The first carbon is here and the second carbon, third, fourth, on fourth we'll have 5th carbon. This ribose sugar helps building a genetic material, that is DNA and RNA. And then deoxyribose sugar. From ribose sugar, we are just going to learn how this deoxyribose sugar is made. C5H10 and O4, it is also a pentose sugar, meaning it has 5 carbon structures, 5 carbon atom. The key difference between the ribose and the deoxyribose is, like you see the structure here, in the second carbon, it is OH. Hydroxyl group will be there in the second carbon, but whereas in deoxyribose sugar will not contain one oxygen here in the hydroxyl group. That is, oxygen is removed. Once when oxygen is removed, we use the term deoxy. Deoxy means removal of oxygen. Where is the oxygen removed from? The second carbon, hydroxyl group of second carbon, the oxygen is removed. And that's why we call ribose sugar into deoxyribose sugar. And next, nitrogen bases. Let's learn about nitrogen bases. Like you see, 2 types of nitrogen bases are there. One is purine, another one is pyrimidine. Purine will be a single cyclic structure that just you can see here that it's a single cyclic structure. It is of thymine, cytosine, and also uracil comes here. Uracil will come here. And then purines will be a double cyclic structure. You can see that there are 2 cyclic structures here. Double cyclic structures, adenine and guanine forms purine. And now, like, easy to remember is pyrimidine, bigger name but with only small structure. Purine, smaller name but with big structure it has. Easy to remember. Then here are purine double cyclic structure and pyrimidine single cyclic structure, cytosine, thymine and uracil. And next, nucleoside. So from nitrogen base and sugar, we are going to now learn about nucleoside. What is a nucleoside? A nucleoside is nothing just a molecule composed of nitrogen base plus sugar. Okay, now look at here. It is, we will learn the difference between nucleoside and nucleotide. The first comes as nucleoside. Okay, the first comes as nucleoside, which is made SIDE, nucleoside, side. Now, the pentose sugar is, obviously we said, deoxyribose sugar builds DNA. Right? And then adenine is a nitrogen base. With adenine, when sugar is added, it becomes adenosine. When with guanine, if sugar is added, it becomes guanosine. Cytosine plus sugar, cytidine. Uracil plus sugar, right, deoxyribose sugar, uridine. Thymine plus sugar, thymidine. Okay, these are 5 nucleotides. Those 5 nitrogen bases along with the sugar forms nucleoside. And for the difference, ribose sugar forms RNA and deoxyribose sugar forms DNA. And next, nucleotide. Let's learn about nucleotide. Like, look at the difference here. It is T. It was side, and here now we are learning tide. A nucleotide is an organic molecule which is the fundamental building block of RNA and DNA. RNA and DNA is a polymer whose monomer is a nucleotide. Now see, what does nucleotide consists of? Now it is phosphoric acid, number 1, pentose sugar, number 2, nitrogen bases, number 3. So these 3 structures, like on the sugar, onto the first carbon of the sugar, we'll have nitrogen base attached, and onto the 5th carbon of sugar, we have phosphate group attached. When these 3 structures combine together, we call it as nucleotide. A nucleotide consists of 3 structures: sugar, phosphate group on 5th carbon, and first nitrogen base on the 1st carbon. This is the image that you've learned in 11th standard. Look at here. This is the double cyclic structure called adenine. When adenine combines with sugar, now this is pentose sugar. On sugar, if adenine is attached, it forms adenosine. This is a nucleoside. They've kept here, see, nucleoside. Nitrogen base plus sugar forms nucleoside. When that is phosphorylated, that is when nucleoside, this is an important question in board also, like, justify the statement that nucleotides are phosphorylated nucleoside. Right? It's correct, no? This is nucleoside. With nucleoside onto the 5th carbon, if phosphate group is added, if nucleoside is phosphorylated, then it becomes nucleotide. Okay, adenosine on phosphorylation forms adenylic acid. This is one of the nucleotide. And next, types of nucleotides. Actually, since we have 5 nitrogen bases, children, we will get 5 nucleotide here. Now, adenine, whose nucleoside is adenosine, that is sugar plus nitrogen base, and here it is, on phosphorylation it forms adenylic acid. Same way, guanine forms guanylic acid. When guanine is found in the nitrogen bases, you call it as guanylic acid. Cytosine, whose nucleoside is cytidine, whose nucleotide is cytidylic acid. Again, uracil, uridine, and uridylic acid. Thymine, thymidine, and thymidyllic acid. So these are the 5 types of nucleic acids we've learned. And next, like, before going into the structure of DNA, let's also learn one more important thing, just a small note. Write it in one corner of your notebook that it is a note, and write at the side as note. Monomer and polymer concept. Look at here, what is monomer? Monomer is nothing, just a single unit. One unit. When single bead is called monomer. When those beads are chained, you call it as polymer. Okay, monomer and polymer. Monomer is a small molecule that are chemically bonded to form a polymer. Some examples of monomer which we have already learnt is here I have kept here. Monomer is amino acid. When amino acids are chained, you get, when amino acids are chained by chemical bonding, it forms a structure called protein. Same way, when sugars, monosaccharides are chained, one monosaccharide is one bead. When you bring many of the beads together, you call it as polysaccharide. That is, it might be cellulose, starch or glycogen. Nucleotide, like what we've learned here is nucleotide. 5 types of nucleotides we've learned, right? Adenyllic acid, guanylyc acid, cytidyllic acid, thymidyllic acid and uridylic acid. Those are nucleotides. When those are chained, we get a structure called nucleic acid. Nucleic acid, nothing, just DNA and RNA. So fatty acid forms glycerol. Next, fatty acid and glycerol forms lipids. Now homopolymer versus heteropolymer. One more a small thing before going into structure of DNA. Here is homopolymer. What is homopolymer? Homopolymer, homo means, the word homo says that it is same. Okay, same. Homopolymer is a polymer composed of same type of monomers. Okay, every single, look at here, every single unit you just check for, it's all the same. When the same type of monomers are joined or chained, you call it as homopolymer. When it comes to, look at this image, like see, monomer unit 1 to 2, 2 is different. 2 to 3, 3 is different, right? Same way again, 1, 2, 3, again, like this is, each single unit is different here. Each single monomer is different here. When that is different monomers are chained, you call it as heteropolymer. Hetero means the very word says it is different. Different monomer units are chained, you will get a heteropolymer. And next, nucleic acid, a heteropolymer. Now we are going to learn, we are into the structure of DNA here. Nucleic acid is a heteropolymer. You look at here, these all adenyllic, thymidyllic, cytidyllic acid, guanylyc acid and uridylic acid actually forms the nucleic acid, DNA and RNA. And now, why do we say that it is a heteropolymer? Now look at here, this unit is adenyllic acid. This unit is thymidyllic acid. Again, one more thymidyllic acid. Again, guanylyc acid. Cytidyllic acid. Again, guanylyc acid. So why we say, since the units are different, each units are different, you call it as heteropolymer. Each nucleotide is different. Different monomer unit forms heteropolymer. You can look at here that adenine, thymine, guanine, cytosine, right? Like it's going, it's chained like that. Now is here a DNA structure. It's, as we have already told, it's a double helix structure. You look at here, it is a helixed structure, helixed in a right-handed fashion, okay, and whose strands are made with sugar and phosphate, whose rungs are made with, in the ladder, the rung is made up of nitrogenous bases bonded by hydrogen bonds. Function, the DNA is actually called hereditary material. DNA is actually called a hereditary material that transmits characters from generation after generation. Components, DNA is made up of 4 nitrogen bases, adenine, guanine, thymine and cytosine. Next, helix to ladder. You can look at here. At first, in the year 1869, when did we know about this DNA? In the year 1869, there was a scientist called Friedrich Mischer. Friedrich Mischer first time identified some acidic content inside the cell. For the first time, in the nucleus of the cell he found, and he only termed it as nuclein. he only termed it as nuclein. And during 1869, 1860s, up till 1900, we were not aware of this DNA structure and all. James Watson and Francis Crick, they introduced the molecular structure of DNA based on the X-ray diffraction data produced by Maurice Wilkinson and Wilkins and Rosalind Franklin. They were even awarded Nobel Prize for Medicine in the year 1962. Now, when this helix structure is like unwinded, you get this ladder model. Antiparallel polarity. Next important thing to learn about DNA structure is antiparallel polarity. Like if you look into DNA is composed of that sugar backbone chain consists of like two strands, two poles in the ladder. If you see that there is in the first strand, the sugar is just kept like that. First carbon, second carbon, third carbon, fourth carbon and fifth carbon. On to the fifth carbon, 5 prime. Now when the sugars are arranged serially like this, this is 5 end here on top where the phosphate group is attached, and 3 prime end where the hydroxyl group is attached. 3 prime end is here. But if you just look into the other strand, it will be anti-parallelly running where you see hydroxyl group, 3 prime end is on top, third carbon is on top, which is attached to a hydroxyl group is in that end when you come downwards, because all it is put upside down. All the sugars and all the content of that strand is upside down. So the fifth carbon will be here. So you call this as 5 prime end. When the first strand is 5 end to 3 end, 5 prime end to 3 prime end, the second strand will be 3 prime end to 5 prime end. And that's why we call it as anti-parallel polarity. Now look at here, the sugars are all upright and the phosphate group for fifth carbon will be up here and the phosphate group will be here. This is fifth carbon. And so you say this one as 5 prime end. Whereas this is into this sugar, so this you call it as 3 prime end. Whereas when you see into the other strand, this is 3 carbon, 1, 2, 3, third carbon. So you say this as 3 prime end and 1, 2, 3, 4, 4 on 5. So you say this as 5 prime end. And next, sugar-phosphate backbone. You just look at here, whatever we learned here. The sugar-phosphate backbone is with sugar and phosphate, makes the poles of the ladder. And then base pairing. The next concept about the DNA structure is base pairing. When we talk about base pairing, here the person who told about base pairing is Erwin Chargaff. This is even called as Chargaff's ratio. We say the amount of adenine, he told us the amount of adenine will always be equal to amount of thymine in the DNA strand, and again the amount of guanine is always equal to the amount of cytosine. Here one purine base pairs with another pyrimidine. Always one single cyclic structure base pairs with the double cyclic structure. The thymine always finds for adenine for pairing. Okay, nitrogenous bases getting paired. That's why you say base pairing. Okay, thymine pairs with adenine. Same way, cytosine pairs with guanine. And one important point to just remember here is always adenine is required with 2 hydrogen bonds. The bond formed between this is hydrogen bond, and the bond, how many hydrogen bonds are required to pair thymine and adenine? It's only 2 hydrogen bonds is enough to pair adenine and thymine, whereas when it comes to cytosine and guanine, it requires triple bond, 3 hydrogen bond. 3 hydrogen bonds are required to base pair guanine and cytosine. And that's what is told here. This is guanine and cytosine with 3 hydrogen bond, adenine and thymine with 2 hydrogen bond. Actually, this is the equation for, this is the equation of Chargaff's rule. A percentage is always equal to T. Adenine percentage proportion is equal to thymine percentage proportion. In the same way, guanine percentage proportion is equal to cytosine percentage proportion. And next comes, here is the put up everything what we have learned, and here let's see the structure of deoxyribonucleic acid. Let us see that the sugar phosphate backbone which makes the 2 strands in the DNA, and then sugar, look at the sugar which we use here is deoxyribose sugar. How do we call it as deoxy? In the second carbon from the hydroxyl group, one oxygen is removed, and that's why we use the term deoxy, removal of one oxygen, and that's why deoxy, these are deoxyribose sugar. Onto its 5th carbon, phosphate groups are attached. And how are these phosphate groups attached? By bond formation called ester bond. Ester bond. Okay, when one sugar combines, like here is one ester bond and here is one ester bond. So you call this as phosphodiester bond. Phosphodiester bond. And again, the bond formed between sugar and nitrogen bases is N-glycosidic bond. The sugar and phosphate, sugar and nitrogen base bonds by N-glycosidic bond, and both the base pairs are bonded by hydrogen bond. Both the base pairs are bonded by hydrogen bonding. And as said, guanine and cytosine 3 bond, adenine and thymine 2 bonds. Now here you see the structure, DNA structure, this unwinded helix, you see that these are backbone, and this is double cyclic structure, and this is single cyclic structure getting bonded. And pitch of the DNA, base pair per turn. How many base pairs? Like see, if you just take the helix to staircase, like say you start here, you just take one full turn and come up here into the straight point. So for this one twist, you will have 10 base pairs. That one twist only is called, one turn is called pitch. One turn is called pitch. For one turn, how many base pairs, how many steps will be there? 10 steps will be there. Your adenine and thymine, like you know, that nitrogen base pairing forms the steps rung in the ladder, right? So those steps will be, there will be 10 steps in it. Now, the distance between the base pairs, from one base pair to another base pair, the distance will be 0.34 nanometer or 3.4 angstroms. Calculating the pitch. The base pair turn, the distance between the base pair turn 10 into the distance between the base pair 0.3 nm forms 3.4 nm or 34 angstroms will be the pitch of one DNA, pitch of the DNA. Now let's learn the difference between DNA and RNA. When we learn DNA is always double-stranded, whereas the difference is RNA is always single-stranded. That you can see that the double-stranded DNA and the single-stranded DNA. Here, adenine, guanine and guanine, the nitrogen bases are cytosine. But see to the difference, DNA is made with thymine, whereas RNA is made with uracil. That makes the difference. The second difference, first difference, it is double-stranded and single-stranded RNA. Second difference is DNA is made with thymine, whereas RNA is made with uracil. And the third difference about it is the pentose sugar. The pentose sugar in ribose is just the ribose sugar. Is just the ribose sugar as such taken, and there is no removal of oxygen there. Whereas here, in deoxyribose sugar, in the second carbon, one O is eliminated. That is the third difference. And DNA dictates the synthesis of protein. Like, protein codes are there in DNA. And from that coding only the proteins are made. So that's why we say DNA dictates for the making of polypeptide chain. And DNA is found in the nucleus. Whereas here, they perform other functions like protein synthesis and all other functions they do. And they are transported. RNAs are made in the nucleus, but they are moved to cytoplasm to carry out their function. But the DNA will remain always in the nucleus itself. Question time. What is DNA? And what does it stands for? What does DNA stands for? What is DNA? DNA is actually our genetic material that inherits characters. What does it stand for? What does DNA? The expanded form of DNA is deoxyribonucleic acid. And what is the structure of DNA? It is a double helixed structure. And then made up of 4 sugar-phosphate backbone with 3 bonds like phosphodiester bond, N-glycosidic bond. Phosphodiester bond connects sugar to sugar. N-glycosidic bonds connects sugar to nitrogen base, and hydrogen bond pairs nitrogenous bases. Next, what are the 4 bases that makes up DNA? Obviously understood: guanine, thymine, adenine, and cytosine. How do DNA base pair up? By hydrogen bonds. Adenine pairs with thymine with a double hydrogen bond, and guanine pairs with cytosine with triple hydrogen bond. Differentiate between DNA. Differentiate DNA and RNA. RNA, as we in the previous page as discussed, DNA is double-stranded, RNA is single-stranded. DNA is made up of deoxyribose sugar, RNA is made up of ribose sugar. DNA will have thymine as one of the nitrogen base. In RNA, thymine is replaced with uracil. Okay, DNA will be in the nucleus always. RNA is formed in the nucleus, but it will be moved to the cytoplasm. Okay, DNA dictates all the protein making process, where RNA helps in making protein. Right? Next, write the equation for Erwin-Chargaff rule. Adenine percentage is always equal to thymine percentage. Guanine percentage is always equal to cytosine percentage. Usually we get questions from this. Like in the board, we'll usually get questions like, if adenine is 20%, what is the percentage proportion of cytosine? Okay, if adenine is 20%, obviously A percentage proportion is always equal to T percentage proportion. So thymine will also be 20, because in one strand if A is there, T will pair up with it. So in that calculation, we say adenine number will always be equal to thymine number. And so if in 100%, if 40% is removed, there will be 60% remaining 2 nitrogen bases. So the 60 divided by 2 will be 30%. If G is 30% and C will also be equal to 30%. That's how we go do with the calculation. They might give you in numbers also. Be curious, stay connected, follow me for more videos. Thank you.